Hi class, we're back in the lab, and today's video we are going to be going over SEM media. So this is in the same exercise as the starch hydrolysis in your lab manual, but they are two different tests. So we have SEM stab, and then we have the starch hydrolysis. So the starch hydrolysis is in a separate video, and this video is simply only SEM stab. Now, the SEM stab is a lot like the motility stab. So the inoculation process is going to be the same. We are going to use our needle and we are going to stab down into the SEM stab because it is a stab just like the motility. SEM media tells us a lot of information about our bacteria. Whenever we're trying to identify what kind of bacteria we could potentially have, the SEM stab is very, very helpful because it can tell us 3 different things about our bacteria. Now, normally, SEM stabs are done on gram-negative organisms because we're looking for intestinal pathogens, so pathogens that live in your intestines. So the 3 different things that we are going to be looking for are motility, tryptophan degradation, and hydrogen sulfide production. So the first part, the motility, SEM stabs have the same amount of agar as motility stabs. They have 1/3 of the normal agar. So that means that the bacteria have the ability to move around. We've already done a motility stab, so we're not going to worry about looking at the motility for the SEM stab, but if you wanted to do that, you could. Second, we can see tryptophan degradation. So, among other things, there's peptone in the media. Okay, there's iron and agar and some other things, but and beef extract. Peptone is going to be our main source. So the peptone is going to provide some amino acids. One of the amino acids is tryptophan. So some bacteria have the ability to break the tryptophan up into pyruvate or pyruvic acid, ammonia, and indole. So the bacteria is able to take that amino acid, break it down into something it can use, and then shuttle it through the Krebs cycle and do respiration. Or whatever else it needs to do, fermentation, whatever. So that's important for the bacteria. So if the bacteria produces the enzyme tryptophanase, which is going to be the enzyme that's going to break down tryptophan, then you're going to produce those products. Now, indole is the product that we are going to test for to see if our bacteria had the ability to break down the tryptophan. The reagent that we're going to add is called Kovacs. So Kovacs reacts with indole and gives you a red color when indole is present. So if our bacteria can break down tryptophan, we add the Kovacs, we're going to get a red ring. If our bacteria cannot break down tryptophan, it does not produce that enzyme, indole will not be present, and when we add the Kovacs, we're going to get a golden/yellow color ring. So red, bright red ring is positive for indole, meaning that tryptophan was degraded. Golden or yellow is negative for that test. So that's the second part of the SEM stab. The third and last part of the SEM stab is looking at hydrogen sulfide production. So tryptophan is not the only amino acid that's in the media. There's also cysteine. And so some bacteria can take that cysteine and deaminate it, and when that occurs, it reacts with sodium thiosulfate that's in the media and produces hydrogen sulfide. Hydrogen sulfide is a gas, however, so we wouldn't really be able to see it. So when hydrogen sulfide is produced, it reacts with the iron that's in the media and produces iron sulfide. Iron sulfide produces a black precipitate. So when you inoculate SEM media and read the results, if you see a black tube, that means that that bacteria had the ability to produce hydrogen sulfide. If the tube is not black, then that means that that bacteria did not have the ability to produce the hydrogen sulfide, so you're not getting that black color. Now, I want to be clear. The black color that you're seeing is not hydrogen sulfide. That is iron sulfide. The black color is caused from the production of hydrogen sulfide, but that's not the black color that you're seeing. The black color that you're seeing is iron sulfide. So why do we care about that? Well, pathogens like Salmonella and Shigella produce hydrogen sulfide. So they are going to produce the hydrogen sulfide, and they're not going to have the ability to break down tryptophan. So they will be positive for hydrogen sulfide and negative for tryptophan degradation or indole production. whereas more common intestinal bacteria will be negative for hydrogen sulfide, non-pathogenic strains, will be negative for hydrogen sulfide and be positive for indole, or positive for the tryptophan degradation. So this helps us to tell the difference between pathogens and then just more common intestinal bacteria. Okay, so now we're going to go over to the workbench. We're going to I'm going to show you how to inoculate the SIM media, and then we're going to read some results that I have ready. Okay, so we're over here at the workbench, and we are going to inoculate some SIM media, and then we are going to go over some results that I have ready. So, like I said earlier, the SIM stab is a stab just like the Motility. So you should already know how to do this, but I am going to demonstrate it for you again. Now, the results that I have are for 3 different bacteria. So I've already done this 3 times for 3 different bacteria. But I'm only going to demonstrate it once for you, because once you do it once, it's pretty straightforward after that. So I'm going to, again, use aseptic technique. So I'm going to flame my entire needle. And remember, we're using our inoculating needle to do the stab. Not going to use our loop. So I'm going to flame the needle and I'm going to let it cool. And the 3 bacteria that I used for this are E. coli, Citrobacter frundi, and Pseudomonas aeruginosa. So that's the 3 results that we're going to read. And we're going to get 3 different results, kind of like we did with the Motility stab. So all you're going to do is you're going to take your bacteria with your flamed needle, and you're going to just glide across the top. Remember, with the needle, you're not stabbing the slant. I'm just going to glide across the top, and then I'm going to stab straight down in the middle, just like you did last time. Go all the way to the bottom, and come all the way back up. And that's it. That's all you do to inoculate your SIM stab. Now, I've got a set that I've already let incubate for 24 hours at 37 degrees Celsius, because that's around the temperature of your body, so that's where they like to grow. Here's our results. So remember, they're threefold. We've got to look at the motility, which we're not going to do because we've already done that before. Then we need to look at hydrogen sulfide production, and then the indole production. So first we're going to look at hydrogen sulfide, because we don't have to add a reagent to do that. We just simply look at the tubes and read the results. So out of our 3 tubes, remember, for hydrogen sulfide production, we're looking for a black color. So out of our 3 tubes, you can see that that middle one is clearly black. That bacteria is Citrobacter frundi. So it's very, very obvious, the black color. Okay, it also smells really bad. So here's my positive reaction for my hydrogen sulfide. Okay, and notice that there's none at the top. It's just all along here on the bottom. So because you want to make sure that it's the butt of the test tube. Okay, so that's my positive for hydrogen sulfide. So Citrobacter frundi is positive for hydrogen sulfide production. Pseudomonas and E. coli are negative for hydrogen sulfide production. Pretty straightforward and easy. Now, the last part of the SIM media is the indole production. So remember, we have to add a reagent to look and see if there was indole produced, meaning that tryptophan was degraded. So the reagent that we're going to add is Kovacs. So there's my Kovacs. And so what we're going to do is we're going to add about 10 drops to each tube, and then we are going to look and see which ones turn red. Because remember, red is going to be our positive result. And these are solid media, so the reagent is going to simply sit on top. And the reason why I say 10, your book says 5 to 10, but you want to add enough to where you can see the ring. If you only add 5, sometimes it can kind of be hard for us to be able to see the ring that we're looking at. So, or looking for. And again, you're wanting to look at the color of the reagent. We're not looking at the color of the media, we're looking at the color of the reagent. So if you look and see, remember, red is positive. So our E. coli is the only one out of these 3 that gave me that bright red color. And a lot of times I like to turn it sideways so that you can see the color of the reagent. So that's that nice bright red. Both Pseudomonas and Citrobacter frundi give me a kind of a burnt orange, dark yellow looking color. And that is negative, okay? So if it's not that, because sometimes this iron sulfide color can, or the iron sulfide itself can make this look like that burnt orange. And then Pseudomonas produces a water-soluble pigment, so it also kind of turns it that orangey looking color. Okay, so even though your book says golden yellow, it's more of like a burnt orange looking color. Okay, so that's a negative result for indole, meaning that these bacteria do not produce tryptophanase and can't break down tryptophan. Okay, and E. coli can. Okay, so that's your results for SIM, hydrogen sulfide production, and then the indole production.